We would like to welcome you to this module entitled Optical Mapping Bacteria of Interest. In this module, we will show you how optical mapping can be a powerful technology for the analysis of Staphylococcus aureus. Optical mapping is a powerful technology to analyze whole genomes of microorganisms quickly and effectively. Without the need of DNA sequence information, optical mapping can be used for comparative genomics, strain typing, and whole genome sequence assembly. Optical mapping does this by using an extremely novel technology that visualizes pieces of genomic DNA. How do we do it? An optical chip, which contains an optical surface and a channel-forming device, is loaded with a DNA solution that flows across the surface. As the solution flows, the DNA molecules attach and stretch across the optical chip. Thousands of DNA molecules attach to the chip. Yes, you can actually see the DNA from one end of the DNA molecule to the other once they are fluorescently stained. However, at this point, there is no way to differentiate one molecule of DNA from another, even though they probably came from different areas of the genome. The optical chip is then processed with a restriction endonuclease that cuts the molecules site-specifically. The cut sites are visualized as gaps in the molecule, and the restriction fragment order is retained because the entire molecule was attached to the optical chip. The option system then marks up a subset of molecules to annotate and labels them in green. The system assesses the size of each DNA fragment of each of the marked up molecules and labels each fragment with its size in kilobases. These are single molecule maps, and this information is specific to the organism that the DNA originated from and is called the map code. The unique map code of all the single molecule maps are compared to each other to find overlapping regions. The single molecule maps are then assembled together to tile across the entire genome. The complete assembly forms a consensus optical map that is representative of the whole genome optical map assembly and contains an extremely unique map code of the organism's genome. Now, this is powerful information. Using Option's Map Solver software, the optical map can be analyzed and manipulated. The optical map contains vertical black lines that represent restriction sites across the entire genome, and the length of the DNA sequence between sites is represented as white space. For more details on optical mapping technology, please watch and listen to our optical mapping overview. The whole genome view of the optical mapping can be used to study organisms that result in deadly diseases in humans and animals. Optical mapping can be used with Staphylococcus aureus to discover genomic rearrangements and modifications to the genomic architecture that other technologies miss and cannot visualize. S. aureus can be typically found in many locations on the human body and normally does not cause any problems. However, methicillin-resistant Staph aureus is a potentially dangerous type that can cause serious infections and death. MRSA infections are becoming more prevalent and have been reported by the media. Option's Map Solver is a highly functional software tool for the analysis of optical map data of S. aureus. Whole genomes of multiple S. aureus isolates can be visualized very easily and at the same time. Here you can see we have a methicillin-sensitive, methicillin-resistant, and a vancomycin-resistant Staph aureus. The similarity cluster function of Map Solver can help you determine if your isolate is the same or different from others. This methicillin-sensitive Staph aureus is 22% different at the whole genome optical map level from this MRSA isolate. Comparing whole genomes of organisms is quick and easy with optical mapping. Areas of similarity are shaded in blue and areas of difference in white. Discover the unexpected. Here's an insertion in S. aureus SB-1 relative to the published MSSA 476 strain. This could be novel genetic information that may be important in your investigation. With an optical map, you get the whole genome's worth of information in not just specific loci. If you are interested in the SEC-MEC region of Staph aureus, you can investigate that just by zooming into that region of the optical map. Optical mapping can even distinguish the types of SEC-MEC cassettes. Here, annotated in gray, is the SEC-MEC region of a methicillin-sensitive Staph aureus. Notice how the map code restriction pattern differs from the isolates below, with the map codes highlighted in blue and red. The blue region represents a type 1 SEC-MEC cassette, whereas the red region represents a type 2. This type of analysis can be done for all of your isolates. With optical maps, you can look at other regions of the whole genome too. With the same optical maps, you can view the SEC-MEC region differences or analyze other regions. Maybe you're interested in prophages and mobile genetic elements. You can zoom in to find those too. Here's the difference that turns out to be prophage col. About 60 kilobases to the right is VSA alpha, and you can see that there is an additional genetic information at this locus in MSSA 476 that is not present in the col strain. Further downstream is another difference that turns out to be prophage Phi SA4. Even further is another difference. This happens to be Phi SA3. Any of these differences could result in a change of phenotype or pathogenesis. Optical mapping truly allows you to view the whole genome. Optical mapping can help you analyze emerging threats like vancomycin-resistant Staph aureus or VRSAs and discover the unexpected. We compared a clinical urine isolate CU-1 to a VRSA isolate and discovered that there is a 273 kb repeated region shown by the red features in the VRSA isolate that would probably never have been detected by other technologies such as microarrays. Here you can see where the map code of CU-1 matches the VRSA isolate. And just downstream, the map code also matches the downstream portion of the VRSA isolate. This duplication potentially has huge implications in the pathogenesis of this VRSA isolate. Optical mapping is a powerful technology for studying Staph aureus. If you are working with Staph aureus, optical mapping is sure to find a place in your investigation. These are just a few examples of what optical mapping can discover. There are many more examples of how optical mapping has been used with other organisms to discover the unexpected. Please contact an Opgen sales representative directly or contact Opgen customer support at 888-85-MAP-IT or customer support at opgen.com to determine how optical mapping can help you.